Today, my guest is Larry Reagan. He's a professor of pharmacology, physiology, and neuroscience at the University of South Carolina School of Medicine. He has done a lot of research on the impact of insulin resistance, diabetes, and bad stress, kind of chronic, uncontrollable stress, psychosocial stress on the brain. And so we'll learn from him about that and his research over quite a few decades now. So welcome, Larry.
Great, thanks, Mark. I appreciate the opportunity to participate today. I've watched some of these other podcasts, and they look like they're a lot of fun.
Yeah, they're a lot of fun. It's a good chance for me to talk with other neuroscientists, and hopefully a good chance for viewers and listeners to learn a little bit about neuroscience, digging down a little bit below the surface even. So are you from Pennsylvania originally?
So I grew up in Philadelphia. I went to college in Philadelphia, and I did my PhD at Penn, and then went to Rockefeller for my postdoc work with Bruce McEwen, and then came here in 2002. So celebrating my 20th anniversary here at the University of South Carolina.
Yeah, that's nice. I've been there once at least that I remember. It's a nice campus. It's a kind of a nice part of the country down there.
Yes, it is.
And so your PhD work, so that was at University of Pennsylvania, and that was kind of a seemingly different than what you ended up doing. It was on angiotensin 2 receptors, is that right?
So we worked a lot on angiotensin 2 receptors, and one of the things I tell the medical students about when I'm talking about cardiovascular drugs with them is that we were among the first groups to study a drug called DUP753, which evolved into losartan, which was the first angiotensin receptor blocker, which is sort of the go-to drug now for the treatment of hypertension. So I did a lot of neuropharmacology, neuropeptides as a graduate student, and that was with Steve Fluharty and the group headed by Alan Epstein at Penn. And then that's when I transferred over to Bruce's lab to do neuroendocrinology and sort of stress, and got into the ideas about metabolic stress when I first joined Bruce's lab. And those sorts of influences were assisted by people like Mary Dahlman, who was also very interested in metabolic stress and how that might impact the central nervous system. So that's kind of where it all started. And, we've been able to keep our stress projects moving and sort of our metabolic disorders projects moving. And I think, as you know, and anybody who's going to watch this podcast know, it's interesting how the plasticity deficits that we see in experimental models and in clinical studies, whether it be in patients who are subjected to chronic stress or metabolic disorders, insulin resistance, obesity, even things like age-related cognitive decline and Alzheimer's disease, a lot of the, shall we say, consequences that we see of those disorders are very similar in terms of structure and function. And there's some common underlying mechanisms, but they're not always the same. And I think that's where we all kind of find our niche, and what we're interested in studying is, am I interested in this particular neurotransmitter system, neuropeptide system, this particular circuit, those sorts of things. But it's interesting that at the end, the structural and functional changes tend to be kind of similar across all of these different types of disorders.
And you and I have both worked a lot on the hippocampus. Well, a lot of people in neuroscience have for a number of reasons, but it's a focus of structural and functional abnormalities in Alzheimer's disease, in depression, in chronic stress. For people who don't know, Bruce McEwen is a kind of a pioneer in studying the effects of chronic uncontrollable stress on the brain and glucocorticoids, cortisol. And yeah, the hippocampus is so important for learning and memory because the circuitry, like all of our sensory information funnels into the hippocampus. And so the pairing of, say, a sight and sound, that information kind of converges on neurons in the hippocampus. And that's, we think, where memories are generally first encoded.
Right.
And so start out by talking, I guess, start with your work with Bruce and stress, glucocorticoids. And I guess even then you were already interested in diabetes and insulin resistance.
Right. So I started a lot of projects as it relates to stress and how we think about chronic stress might be an important component to neuropsychiatric disorders. We did some studies, with stress and plus and minus different types of antidepressants, looking at structural changes and things of that nature, including oxidative stress, which was one of the things where you and I overlapped a long time ago. And so during that same time, though, it became really clear, again, with studies from people like Mary Dahlman and other folks, including Alex Di Nicola, who was in Argentina, that if you looked at animals with metabolic disorders, they had impairments in HPA axis function. And, an animal with poorly controlled type 1 or type 2 diabetes, those animals had elevated glucocorticoid levels. So, again, sort of emanating from a lot of studies from Mary and other people, it came up this concept of, metabolic stress. And that's where we started. We did a lot of studies back in the day with type 1 models, which were popularized by the use of streptozotocin to, destroy pancreatic beta cells. There's some advantages to those types of studies. I mean, I think the major disadvantage is that you're talking about a uncontrolled hyperglycemia. I would say super physiological elevations in plasma glucose levels. And so the question then becomes how relevant is that to the clinical setting? And so as a result, we started shifting more to type 2 diabetes models or obesity models, sort of like the DB mice, which I know you've worked with, and OBs or Zuckers. And I guess another sort of pioneering team as it relates to changes in the central nervous system in a Zucker animal or obese animals were Carol Greenwood and Gordon Winokur. So they showed in the late 1980s that animals which were provided a high-fat diet exhibited deficits in hippocampal dependent learning and memory. And so, that started a lot of work from many different laboratories. And we were able to, set up a collaboration with them when I was still at Rockefeller, and sort of during that transition time here to USC, where we did very similar types of studies in the Zucker animal, showing that they had deficits in hippocampal dependent learning and memory. And we also showed that those animals had insulin resistance in the hippocampus.
And that,
go ahead, Mark.
And I should just mention, you mentioned DB and OB. So these are DB mice have a defective leptin receptor, and OB don't produce leptin. And people with obesity commonly have leptin resistance, and actually kind of similar to insulin, very similar. They have elevated leptin levels, but their brain isn't. Leptin is the I'm full, stop eating, you've eaten enough signal. And this is very common. I've seen this a lot just out and about at restaurants or whatever, waiting in line. People with obesity, they tend to perceive they're hungry a lot, more so, I think, than people of normal weight, and so once they have obesity, they tend to keep overeating because that leptin system isn't telling them, okay, you're full, you've eaten enough.
Right, and that's a great point. It beyond the DBs and the OBs, you have the same sort of thing with the Zucker animals, is that they have deficits in their insulin or their leptin receptor signaling. And so that's a perfect segue into one of the things that we became very interested in is that what's the role of insulin? What's the role of leptin? And as it relates to plasticity deficits. And so, going way back in time, we know that there was a period when people really felt like the central nervous system didn't respond to insulin. It was considered an insulin insensitive organ. And I actually remember going to meetings in the 1990s where people would stand up in the meeting and say that insulin doesn't work in the central nervous system. And sometimes you felt like standing up saying, I'm here. No, that's not true. But anyway, so you're right. And one of the things that we've become really interested in is sort of disentangling what role insulin might be playing, what role leptin might be playing, which is why when we first got here, my team first got here, one of the things we were very interested in is more selectively targeting insulin receptors in the central nervous system. Because as you've noted, when you're talking about obesity, there's lots of endocrine changes which are taking place. The patients have elevated leptin. They're not responding to leptin. They have insulin resistance. There's changes in triglycerides, changes in cortisol levels. They have increases in adiposity. They have increases in inflammation. And so one of the things that we became very interested in is, are there ways that you can more selectively identify how insulin might be participating in these sorts of deficits that you see? Because again, if you look at the clinical setting, it's not just those endocrine changes, it's the consequences of those endocrine changes. Again, cognitive deficits. You have enhancement or an increased risk of neuropsychiatric disorders, age-related cognitive decline. And then, if you go underneath that in terms of the molecular mechanisms or the cellular mechanisms, it's oxidative stress, like we've talked about. It's structural changes. It's changes in receptor systems. So again, it's sort of like the layers of the onion, which might be consequences, but there's also layers to the onion in terms of what the causes might be. And so that's why we started trying to target insulin receptor populations in different parts of the brain.
So neurons definitely have insulin receptors. And does insulin cross the blood-brain barrier, or is there insulin produced by cells in the brain?
So that's a good question. Let's start with the easier one. The easier question is, we know for sure that peripherally derived insulin crosses the blood-brain barrier. Some very pioneering studies from Bill Banks and his colleagues, where they could show using radio-labeled insulin that it crosses the blood-brain barrier. Mechanistically, how that takes place, I think, is still unclear. One of the hypotheses is that there's an insulin receptor expressed at the blood-brain barrier, and that when it binds insulin, it actually pulls insulin across the blood-brain barrier. So that's sort of the ongoing hypothesis about how insulin, which is released from the pancreas, gets into the central nervous system. There are also investigators who have provided evidence that insulin is also made in the brain. I don't know. I think that that's something which is somewhat debated in the field. But I think irrespective of the source of insulin, we know that it's in the central nervous system, and it can bind to these different receptor populations throughout the central nervous system.
Now, you did some nice studies where you used you essentially selectively knocked out the gene encoding the insulin receptor only in the hippocampus. And those are really kind of very important advance in the field, kind of nailing down that actually the resistance of neurons to insulin is sufficient to cause some of these abnormalities in cognition or synaptic plasticity. So could you take some time and talk about those experiments?
Yeah, sure. Like I said, when we first got here to South Carolina, one of the things that we were interested in, again, is doing this more targeted approach. And so when we developed this insulin receptor antisense sequence, we put it into a lentivirus, which back at the time was, the lentivirus approach was sort of cutting edge, sort of not as much anymore. But one of the things that we were interested in, and a lot of my colleagues suggested, is that you need to validate the efficacy of this virus. And in fact, this was something which was suggested by the study section panel. And so we did those experiments, and we targeted the hypothalamus. And when we targeted the hypothalamus, we got the sorts of changes that you would expect, which was we had increases in body weight, we had increases in adiposity, increases in plasma leptin levels. And so that led us to a whole other series of experiments where what we were looking at was how those deficits in insulin receptor expression in the hypothalamus impacted the hippocampus. And so hippocampal insulin receptor signaling and expression was fine, but when we looked at plasticity measures, when we looked at long-term potentiation, when we looked at structural changes, when we looked at behavior, all of those parameters were reduced in animals in which we downregulated hypothalamic insulin receptors.
Okay.
So this kind of recapitulated a lot of the data, again, data that you contributed to in terms of showing that an obesity phenotype impairs hippocampal synaptic plasticity. So what that started to let us do is begin to identify what the causes might be of those deficits in hippocampus, including, for example, leptin resistance. But when we fast forwarded those analyses to say, okay, great, we know that that sort of recapitulated a lot of the work that we see in obesity studies. What's the specific role of what's happening with insulin receptors in the hippocampus?
Yeah.
And so then we basically moved our target from the hypothalamus to the hippocampus. And what we were able to demonstrate in those animals was, again, we saw deficits. If we produced a hippocampal specific insulin resistance, we saw deficits in long-term potentiation. We saw changes in glutamate receptor subunit expression and phosphorylation and trafficking. We saw structural changes. We saw decreases in neurogenesis. And importantly, we saw decreases in hippocampal dependent learning and memory. And we also saw more selective depressive-like behaviors in those animals. So we saw, interestingly, we saw decreases in, in the appearance of behavioral despair in the forced swim test, and we saw anxiety-like behaviors. But we didn't see any changes in sucrose preference, which suggests, not surprisingly, that's probably more involved in areas related to motivation and reward and things like that. So what that really identified for us was, here's another interesting thing. In those animals in which we specifically target the hippocampus, we see that they have decreases in BDNF. And so what this suggests to us is that there are a series of causes of insulin resistance, say inflammation, oxidative stress, dysregulation of the HPA axis, all of which may produce insulin resistance. But then in terms of what the outcomes are, it's all those hippocampal synaptic plasticity deficits that we see, and including things like decreases in neurotrophic factors. So it's allowed us to sort of identify and differentiate between causes and consequences. That's one of the major areas as we've been looking at.
Okay, so talk a little bit about the insulin receptor itself, and how does it signal downstream into the cell?
So getting back a little bit to that idea about the brain being insulin sensitive, and we know from, again, the 1970s that we can see insulin receptors in the central nervous system, whether that be messenger RNA or binding activity or things of that nature. And one of the things that we know is that there's a developmental regulation of those receptors, and there's also important time windows as it relates to how insulin might mediate plasticity across the lifespan, and that might be something else we want to chat about as it relates to things like Alzheimer's and aging. But the receptor itself is a tyrosine kinase receptor, a classic tyrosine kinase receptor, similar to, say, the leptin receptor. And one of the first things that happens when you get insulin binding to the receptor is you get autophosphorylation of the receptor, which then recruits insulin receptor substrate proteins, which then leads to the recruitment of other adapter proteins, and ultimately can go on and lead to the activation of two sort of independent signaling pathways. One is the AKT pathway, and the other is the MAP kinase pathway. And one of the really interesting things that we, not only we, but a lot of other people have shown, is that you can sort of distinguish between how that pathway is activated, especially at the level of IRS1 proteins and AKT, and also ways that that system can be turned off, especially as it relates to, again, things like Alzheimer's disease. So again, it's really important in differentiating between the activation of that pathway and how that pathway may be disrupted. One of the things that we have shown in our studies is that activation of that pathway leads to translocation of GLUT4 from the cytosol to the plasma membrane, just like has been shown in peripheral tissues. And so I think one of the things that we've talked about over the years is that insulin receptor activation can lead to maybe transient and localized increases in glucose utilization, glucose uptake and utilization. Our colleague Ewan McNay has done a lot of studies to support this idea as well. And because he's actually shown that you can get these localized decreases in glucose, localized increases in glucose uptake with insulin receptor stimulation. And so I don't think that insulin per se plays a major role in glucose metabolism like it does in the periphery, obviously. But I think under these situations in which you have this increased demand for glucose or increased demand for an energy source, say during periods of high plasticity, this might be a way to provide that metabolic fuel, this translocation of glucose to the plasma membrane. And then obviously, beyond that, we know that there are changes in gene expression and things of that nature.
So GlutFlow, glucose transporter 4, as you mentioned, that's like the classic one in their muscles and liver. Insulin, when insulin binds the receptor, then that causes more of the glucose transporter to be inserted in the membrane, so the cells are better able to take up glucose. We did some work on GLUT3, which is another glucose transporter that seems to be, seems highly expressed in neurons relative to, but it doesn't seem to be responsive to insulin. It's kind of constantly there taking up glucose. Now you mentioned BDNF, and pretty much all the effects you've seen with when you knock out the insulin receptor in hippocampal neurons, impaired learning and memory, reduced LTP, which is strengthening of synapses, and morphological changes, impaired neurogenesis, they could all in theory be accounted for by a reduction in BDNF levels. Has anyone looked whether insulin, if you activate insulin receptors in neurons, does that alone induce the production of BDNF?
So that's a great question. And the answer is I don't know the answer to that. I would assume if there are changes, it would be something like changes in, say, message, which then leads to changes in protein. I know that, again, if you're talking about hyperinsulinic states, insulin resistance, or in our model where we have this targeted insulin receptor decrease in hippocampus, in all of those settings, you see this decrease in BDNF. And we found that both in plasma as well as in the hippocampus. And I think what's also interesting, and I'm thinking about this other podcast that you did with Art Kramer, where you talked about exercise. And I know that the both of you have done a lot of work in exercise, and one of the hypotheses as it relates to the pro-cognitive effects of exercise is mediated through BDNF. Right. And so that's another area that we're interested in looking at, is in terms of exercise and how that might promote plasticity. Is there a way that exercise can overcome this insulin resistance? And one of the endpoint measures that we'll be interested in looking at is how might that promote increases in BDNF levels, not only in plasma, but also in hippocampus.
Yeah, that's straightforward experiments. You can do the same, knock down the insulin receptor in the hippocampus and then exercise animals and see, whatever the result is, it's interesting and important. It would be pretty remarkable if exercise was no longer able to, either way, no matter what the result is, I think it's would be pretty important. You could also do intermittent fasting.
Right. Yes. Indeed. So we have done that. I think a lot of folks have done studies like that too, and they've shown that in obese animals or animals provided a high-fat diet, that if you change the diet or food restrict the animals, that you can restore hippocampal plasticity. Yeah. So this is another experiment that we've never done, which I've always wanted to, which is we know that in the periphery that exercise promotes insulin-independent glucose uptake. And so one of the things that we've thought about is, well, what might be happening in the central nervous system? Is there also insulin-independent glucose uptake with exercise? And I think there's evidence to support that. I guess the question is, mechanistically, how does that take place?
Let's talk a little bit about aging, mild cognitive impairment, Alzheimer's disease, and insulin resistance. So there's quite a bit of epidemiological evidence that people who are chronically insulin resistant during their adult life are at increased risk for age-related cognitive impairment and Alzheimer's disease. And you mentioned that with obesity metabolic syndrome, essentially. A lot of belly fat, white fat. It's just thought of as a kind of a bad fat, a pro-inflammatory fat, and insulin resistance. So these all lead to increased oxidative stress. And oxidative stress, we did a lot of work on this actually in relation to Alzheimer's disease, initially in cell cultures and then in animals. And so it turns out there's a lot of proteins in the membranes of neurons that are very sensitive to oxidative stress. I mentioned the glucose transporter 3, which we found if we subject neurons to something called membrane lipid peroxidation, which is kind of free radical attack on the membranes, then that can, through the certain mechanism, impair the function of glucose transporter, sodium pump even. And we didn't look at insulin or insulin-like growth factor receptors. But nevertheless, you have, so in Alzheimer's disease, if you look at the brains, they have a big increase in membrane lipid peroxidation, oxidative stress. And so you want to talk a little bit about what's happening during aging and in Alzheimer's disease in terms of your thoughts on alterations, impaired glucose metabolism, potential role of insulin resistance, and also some clinical trials work that suggests a potential that if it's possible to increase insulin sensitivity, that could be beneficial.
Yeah, so that's a lot of areas. So let's get started. So I think one of the things that we talk about, again, in terms of across the lifespan, is that we know that one of the things that we talk about is that, beginning at the earliest stages of development and to midlife, we know, I like to say that insulin provides a substrate upon which plasticity can take place. So it promotes plasticity in the context of sort of providing a scaffolding for plasticity to take place. But I think as we age, we start to see that now insulin resistance becomes problematic in neurodegenerative disorders, neuropsychiatric disorders, and so forth. And that can be exacerbated when we think about individuals who have insulin resistance or obesity, type 2 diabetes, the metabolic syndrome. And that a lot of investigators refer to this as accelerated brain aging. I mean, I know that you've used that term as well. And the term that we worked with from the McEwen lab was this allostatic load. So this combination of aging along with a metabolic disorder, again, would exacerbate the consequences of aging. And so there have been a lot of studies which have shown that if you look in postmortem brains of Alzheimer's patients, you see that there are decreases in insulin receptor signaling. And so people like the Arnold group and the DeFelice group, folks like that have shown that in postmortem studies, maybe not so much about the receptor expression itself, but if you look at signaling, those downstream signaling, whether that be the phosphorylation of AKT or the phosphorylation of those insulin receptor substrate proteins, that they're all reduced in patients with Alzheimer's. And interestingly, those deficits are correlated with the degree of cognitive impairment that that individual patient had. So again, it would suggest that, and again, getting back to this idea about cause versus consequence, I don't necessarily think it's a cause or a consequence. I think of insulin resistance as sort of being a keystone feature between the causes and the consequences. And so I think it's pretty clear that patients with Alzheimer's disease have insulin resistance in their brains. And obviously, one of the things that we think about in terms of that cascade that we talked about earlier, that signaling through insulin receptors, is that when you get it under normal circumstances, when you get activation of the insulin receptor, you get the GSK3 beta gets turned off. And we know that that's regulating the phosphorylation of tau. So if you have insulin resistance, conversely, that means GSK3 beta levels or activity go up, which is now you're going to getting hyperphosphorylation of tau. And now that could be one of the major histopathological features of Alzheimer's disease. We also know that there's sort of this relationship, this vicious cycle that we think about in terms of beta amyloids and how they impact insulin receptor signaling and how that might ultimately impact IDE or insulin degrading enzyme, which then further feeds back to impair insulin signaling. So there's a lot of evidence to suggest that insulin plays a really key role in leading to those histopathological features that we see in Alzheimer's. Now, as it relates to some of the therapies, obviously, one of the things that we think about is whenever we teach our medical students, right, Dr. Madsen, we always tell them first about lifestyle interventions, right? So diet and exercise, and I know you've done a lot of the work with exercise, and you talked about that with Art Kramer. But you know, in terms of other strategies, a lot of the other strategies, pharmacological strategies, have been, shall we say, somewhat disappointing. I think part of that has to do with this, the earlier the intervention, the better.
Yeah, for sure.
I think that if you look at any disorder, the earlier that you intervene with the patient, the better, whether it's asthma, whether it's COPD, whether it's GI disorders, central nervous system disorders. The sooner you intervene, the better. And I think the sooner you intervene, the more likely a lot of these drugs that we have might be more efficacious. If somebody's already in Alzheimer's disease, it's sort of like the horse is already out of the barn.
Yeah.
As it relates to the strategies that we're referring to enhance insulin receptor activity in the central nervous system, one of the strategies which is gaining a lot of traction is intranasal insulin administration. So from a historical perspective, you know, Bill Fry was somebody who was very interested in intranasal administration of drugs. You bypass the blood-brain barrier. You don't have effects on peripheral systems. You avoid the first pass effect. So it's a great strategy, and one that we use all the time. We're using here in South Carolina right now to avoid our allergies, as you know, the nasal administration of drugs. A lot of the early studies as it relates to sort of the physiology of that were done by Christian Benedict and Paul Schmidt and his colleagues, and they showed that in healthy young adults, they could increase both mood and cognition without impacting peripheral glucose homeostasis. So those sorts of studies served as the groundwork for studies that people like Suzanne Kraft are doing now, where they're showing, there's been a lot of really elegant studies which have shown that if you administer intranasal insulin, maybe not acutely, but chronically, you get enhancement of cognitive function in patients with Alzheimer's disease. But what's also interesting, we know that, you know, ApoEs play a big role in all of this. And one of the things that they've shown is that ApoE4, which is considered to be one of these risk genes for Alzheimer's, that those ApoE4 patients, depending upon the formulation, they may show cognitive enhancement, or at the very least, you sort of stabilize their cognitive function. So let's think about it. Even if we could stabilize the cognitive function of a patient with Alzheimer's disease, that would be a huge help, huge help. Not only for the patient, but let's not forget the major stress that it provides the caregivers.
And there's been advances in earlier and earlier diagnosis too, you know, based on a combination of cognitive testing and biomarkers. And there's a lot of effort. You know, there was a neurologist in my group at NIA, Dimitrios Kapagiannis,
Yes.
who you'd probably come across, I would think. And so he's been doing a lot of work with what are called extracellular vesicles or exosomes, which are these very tiny membranous vesicles that apparently all cells release. And so he's been able to, using antibodies against a neuron-specific protein, to first you take blood sample and you essentially through centrifugation or other means isolate these small vesicles, all of them. Then you take an antibody against the neuron-specific protein and use it to pull out the vesicles that we think are probably coming from neurons. And he's shown that, you mentioned this IRS protein, insulin receptor substrate, that's involved in the signaling and its phosphorylation state, and that can be looked at using different antibodies. But anyway, bottom line, he found in vesicles from Alzheimer's and mild cognitive impairment patients. So he pulled out the vesicles and found they had impaired profile of the IRS2 protein. So that's all consistent with your work and others earlier. But I guess my point is that's potentially one early biomarker in addition to looking at phospho-tau, for example.
Right.
And
Those
so,
I'm sorry, go ahead.
yeah, so you can start the trials in people earlier and then ideally follow them over many years. That's another thing with the clinical trials. Oftentimes they're fairly short, 6 months or a year. And, even a year is, it's a meaningful amount of time, but in the big picture, it's not. Yeah, so there's a lot of issues with the clinical trials. I also wanted to mention, and we can talk briefly about this, other approaches to improving insulin sensitivity. We did some work, just by chance, when I moved to NIA, Josephine Egan, who's a diabetes expert, and Nigel Gregg, who was in my group when I came to NIA, they were working on GLP-1, glucagon-like peptide 1. I'm going to have a podcast on that later with Tim Foltini, who's been doing clinical trials in Parkinson's patients. But anyway, so when you eat, there's a protein released from cells in your gut into the blood. It's called glucagon-like peptide 1, GLP-1. And it does several things that are good for glucose regulation. It stimulates insulin release from the pancreas, but it also acts directly on muscle and liver cells, and we think neurons, to enhance their insulin sensitivity. So that's potentially another approach. It does have a potential downside, and well, it's actually an upside for people with obesity. This has been in the news a lot lately.
Yes.
And this was first developed by Josephine Egan and Nigel Gregg at NIA. These GLP-1 called analogs. So they're modified forms of GLP-1 that stay around longer in the blood. And the first one was exenatide or Exendin-4. And that's widely used in people with diabetes. But now you see in the news, these same drugs they're being prescribed now like crazy for people with obesity. I'm not sure this isn't the best approach in my view. Exercise, reducing calorie intake, that works really well. So far, these drugs look pretty good as far as side effects. But one side effect, it's actually the effect that's good for people with obesity. They inhibit appetite.
Right.
So the people don't feel hungry. But with Alzheimer's, I don't know. This is kind of a tricky business because Alzheimer's mainly affects people in their 70s, 80s, right? 80s is typical. And so, they may be losing weight already, not keeping their nutrition. Demetrios Kapagiannis actually did a small clinical trial of Exendin-4 in patients with early Alzheimer's. I'm on the paper. I didn't really do much. I helped a little bit, but it was a small study, short time period, 6 months, and he didn't see any effect on cognitive outcomes. That doesn't mean, it's a small study. That doesn't mean this should be pursued further. Anyway, so.
Well, and it's interesting you talk about, biomarkers and those exosomes. I mean, that would be a great strategy if you could show, for example, in a patient early on that they have these changes or certain types of phosphorylation in IRS2, and now you begin the intervention, diet and exercise, maybe some of these drugs, intranasal insulin administration. And now in that same patient, you could follow that phosphorylation of IRS2 over time and correlate that with their cognitive function. And then I think that would give you sort of a window to the brain and what's happening in those patients. Yeah. Are you restoring insulin signaling? Are you overcoming insulin resistance? Those are the sorts of biomarkers that we need to really tell us you know, as we're going through these clinical studies, are we seeing positive outcomes?
Yeah.
In terms of the incretin analogs, there have been a couple other clinical studies which have been done. One of which was by my colleague in the Netherlands, on Beasels, and I think some of the outcomes have been disappointing. And again, one of the things that I think about is, would those outcomes have been more successful if we had started earlier? I mean,
Yeah.
This is I think at some point with a patient with a neurodegenerative disease, it's going to be really difficult to overcome any of the changes which have already taken place.
You know, Larry, this is something that, yeah, I agree 100%. And a lot of times the way animal studies are done, you know, typically any drug development, you start in animals.
Right.
And so with these neurodegenerative disorders, whether it's ALS, Alzheimer's, Parkinson's, the animal studies, they generally, you give the drug before or at a very early stage. You know, for example, ALS model, where the animals will have degeneration of motor neurons in their spinal cord and progressive paralysis, and you can evaluate that by looking at their motor function and so on, and strength, muscle strength, essentially. And in the studies I'm aware of, the vast majority, they give the drug before the animal starts to show any motor dysfunction or even pathology. And so they're more like prevention or delaying the onset of the disease. And then, whereas the clinical trials, you're giving it to someone who's already, you know, process. So I think that's another big caveat in the translation, the way the studies, animal studies are designed.
And I completely agree. I mean, I think when you compare the animal studies with the clinical studies, that's what we see, is that it's too late. So I think one of the things that we can think of from a public health perspective is this further emphasizes the need for healthy lifestyles.
Yeah.
And I don't think that means you need to be, you know, someone who's doing ultra marathons, right? It's even just moderate exercise, having a moderate lifestyle in terms of, you know, what you eat. I think that just that, if you're slowing the progression of that natural aging process, or, you know, we know this, that we still know that as a result of the normal aging process, that there's shrinkage of the hippocampus and that there's modest cognitive decline. So if you could slide into that phenotype versus having a situation in which you have other pre-existing conditions, metabolic syndrome, hypertension, cardiovascular disease, whatever the case might be, that's going to slide you into that phenotype which goes into MCI or AD, that's what we need to do, is intervene at those time points and even perhaps start preventative measures in patients, because that's what the animal literature is telling us. So I think we talk about this bi-directional relationship in terms of translation. The animal studies are telling us maybe start earlier, and maybe that's something that the clinical studies also emphasize, that if we start too late, the interventions aren't going to be very effective.
Yeah, we're still at the point, in my view, that people shouldn't expect a drug to be effective in someone who's already, you know, probably even when they're already starting to have clear cognitive problems, there's already a lot of bad things going on. This is something I've talked about. I finally got around to writing a book on the science of intermittent fasting.
Right.
And I put a chapter in there on the dark forces and the kind of the way the whole system's set up, our whole healthcare system in this country. And we're like maybe the worst example of how to go about healthcare, right? In de-emphasizing or not even emphasizing at all risk reduction and prevention, or putting a lot of resources into that. So we're mostly geared for letting people get sick and maybe even facilitating, you know, processed food industry, pharmaceutical industry, even, you know. I think a lot of people in this country expect there'll be some drug that's going to help them. But and then the other thing is, this gets into like a transgenerational scenario where if a child is born and their parents have really bad health habits, they're sedentary, they eat junk food all the time, they don't keep their mind intellectually challenged by whatever, reading book or things, then the kid's more likely to have that lifestyle and those bad habits when they grow up. And so it just kind of, you get this snowball effect. There's a big increase in childhood obesity, right? And you and I would think, we're definitely worried about that, because even in kids, there's evidence that, and I've seen, and you may know more about this than me, but I've seen multiple studies where they do cognitive testing on children with obesity and their peers, their schoolmates who have normal body weight, and the data seem to show that on average, kids with obesity perform more poorly on cognitive tests. There's been even some brain imaging showing small but statistically significantly smaller hippocampus, even in children with obesity. So their brains are clearly at big risk as they get older.
Yeah, and beyond the cognitive performance, there's also some of those studies suggest that there's also this inherent risk in that population to spend more time in the principal's office or more time in trouble outside of school. And so there's a lot of factors that go into that, Mark, right? I mean, a lot of times that comes into, socioeconomic status. And one of the things we think about is the individuals, the families that live in certain environments where they have food deserts. They don't have access to a good supermarket, and they're eating food that they get at the local pantry, at the gas station, things of that nature. And so SES plays into this as well.
Yes.
And so now you have a situation where those kids are already fighting an uphill battle. It's almost like their accelerated brain aging has started much earlier in life for them. And again, I think this goes back to your point, the work that you've done is it really emphasizes the importance of healthy lifestyle and how that can impact brain structure and function.
Yeah. Yep. Okay. Let's see. I had a couple. So you talked a little bit about glucocorticoids, cortisol, and its bad effects on the brain. And let's dig down a little bit on that. I did have Robert Sapolsky on way back on one of my, and we did some collaborate when I was at University of Kentucky. I had a graduate student who collaborated with Jim Herman. He probably, he was at Kentucky then. Now he's at Cincinnati, and so there was this kind of paradoxical thing where if we take rats or mice and we do daily calorie restriction or every other day food deprivation, every other day fasting, if that's begun when they're young, they'll live a lot longer than the control animals, with the caveat that the control animals they're actually probably overeating, definitely compared to in the wild. But nevertheless, these animals with caloric restriction, and we did this with intermittent fasting, they have elevated glucocorticoid levels. In the case of rats and mice, it's corticosterone, but it's the equivalent of cortisol. And so there's this it's kind of paradoxical, because generally you think of high cortisol levels as being a bad thing. You mentioned with obesity, type 2 diabetes, elevated cortisol levels, chronic stress. So we wondered what's going on there. And we were seeing good effects of this intermittent fasting on the brain, even though they had elevated glucocorticoid levels. So Jim collaborated with graduate student Jaewon Lee in my lab, and we essentially measured relative levels of the two different receptors for cortisol. One's called GR, glucocorticoid receptor. The other is MR, mineralocorticoid. And previous studies with kind of work Bruce McEwen and others have done, you have kind of chronic psychosocial stress. We could talk about the way this is done in the lab, but essentially you semi-terrorize the animals, I guess, or, everything from sleep deprivation to like putting them in cold water, whatever. And if you do that chronically, it's bad for their hippocampus. But anyway, in the bad stress scenario, levels of MR are reduced and GR stay up. And we found the opposite occurred with the intermittent fasting. MR stayed up and GR went down a little bit. So, which I think that was actually an important finding. I haven't seen people follow up on this much, but so our thinking was that the way the neurons are responding to the cortisol is different because it's the way these two different receptors signal and probably the gene targets. I should say these receptor proteins are transcription factors, so they will affect gene expression. So what do you think about that? That's a pretty interesting finding, I thought.
It is really interesting, and I think one of the things to keep in mind is, and I'm sure your studies did this, is that there's a difference between the effect of activation of the HPA axis in response to a stressor, an acute stressor, and we can talk about those different types of stress paradigms, whether they be, chronic restraint stress, which is one we've done a lot, or a lot of folks like Jim's lab, they do these unpredictable stress paradigms so that the animals don't habituate to the stressor. And so every day they see a different type of stressor. But I think the key is that in those settings, at least at the early stages of those chronic paradigms, you see that there's a big spike in plasma levels of glucocorticoids, which then come back down to baseline. And then as the animal goes through those paradigms, that effect may or may not be blunted, depending upon the type of paradigm that you're looking at, right?
Yeah.
And so you might see this increase, and then it comes back down. Whereas perhaps what you're seeing in your studies, and we've also done food restriction studies, and we haven't seen changes in plasma glucocorticoids, but the food restriction that we did was much more mild than what you did with your studies, and in combination with Jim. So ours were maybe they got about 80% of what their normal daily food intake was. And I think you guys were doing like, they were getting what, 20 to 30% of their normal food intake?
30 to 40%.
30 to 40%. So I think in that situation, that might have been maybe a very mild chronic stressor, which perhaps over the 24-hour cycle, you saw perhaps and not as big an increase as it relates to what you would see with a stress paradigm. Right. And so under those circumstances, now if you've got a shift in the expression of those various receptors, again, just like you said, that might lead to differential changes in gene profiles, which if you're thinking about chronic stress, those gene profiles ultimately lead to deficits of plasticity. Whereas perhaps in the setting where you're looking at, whereas now you're getting a greater activation of those mineralocorticoid receptors versus the glucocorticoid receptors, maybe that's now promoting plasticity, promoting resilience because of certain genes which have been upregulated. And that's, I don't know if you guys did any gene profiles on that, but that's really interesting to look at, because I think now if you looked at those gene array following those two different types of paradigms, I suspect that there would be very, very different gene profiles. And in the intermittent fasting, you might see something like where the genes which are being upregulated are more related to resilience, whereas in a chronic stress paradigm, it's maybe an increase in the genes that might be related to vulnerability. For example, maybe there's changes in BDNF that you would see in intermittent fasting that you would clearly know as it relates to chronic stress. We see decreases in BDNF.
Yeah, and Bruce had shown, right, that glucocorticoids decrease BDNF, but we did follow up a little bit. Alexis Stranahan, you probably bumped into her. So she was a graduate student with me and did all her PhD work in my lab. She got her PhD at Princeton.
Yep.
NIH has this, I don't know if there's any potential graduate students listening, they may be interested in this. The NIH has a program that's called the Graduate Partnerships Program. So they have formal arrangements with Oxford and Cambridge, with the Karolinska. I've had a student from there, and from Princeton, where Alexis was. But anyway, so Alexis, you know her work because she did stuff in DBD mice, taking out the adrenal glands, the source of the glucocorticoids, and then seeing beneficial effects on the hippocampus of removing the glucocorticoids. But she also did a study where she used aldosterone,
Right.
which selectively activates MR,
Right.
and found that, and I should remember this, I can't remember which model it was. I think it was either diet-induced obesity or the DB mice that seemed to have some beneficial effects.
Right, and that would go back to this whole concept in terms of the differential occupation, right? Because we know glucocorticoids bind to both of those receptors, and we know there's the circadian rhythm of glucocorticoids. So now, depending on the time of day, you're going to have differential occupancy of MRs versus GRs. And now, if you're talking about a situation where you're driving MR activation, that could be recapitulating what you're talking about in terms of your intermittent fasting. Yeah, it's very interesting.
Now you got me thinking about sleep. What's going on during sleep with?
So yeah, and so that's a great question. And one of the things that I listened a little bit to the session you had with Ted Abel as well, and in terms of the how we get that migration that he called it, migration of our memories from hippocampus to those cortical areas, and how we think that sleep might be involved in those processes, but. When we think about our chronic stress paradigms, doesn't matter what paradigm you use. If you use unpredictable stress, use chronic restraint stress, whatever it might be, we're always doing that during the light cycle because that's very convenient for us as scientists, as investigators. But let's keep in mind, rodents, that's when they're supposed to be sleeping. So some people have even noted that perhaps what we're looking at with chronic stress is, sure, it's an effective activation of the HPA axis. You get habituation of the HPA axis and so forth, but maybe some of it's also related to the fact that we are disrupting the sleep patterns in animals. And I know there are a lot of folks that are looking at that, and I'm not an expert in that, so I don't even want to speculate on what the potential outcomes could be or mechanisms could be. But I think that has to be a component of it when we think about chronic stress in animal studies. And if we think about in the clinical setting, if somebody has PTSD, if someone has depressive illness, if someone has mild cognitive impairment or Alzheimer's disease, one of the common features that we see in all of those is disruption in sleep patterns.
Yeah.
And so I think that, again, getting back to where we started, I think that there are potential common causes and common consequences, but how we get there might be different. And here's an example of maybe sleep disturbances is a perhaps both a cause and a consequence in some clinical settings.
Yeah, the sleep Americans, a lot of them don't get enough sleep, and that alone is a stress, as you kind of mentioned. And so, yeah, people should try, do whatever they can to get, 8 hours of sleep at night, and because it's so important. But sleep deprivation, so people with anxiety disorders often, chronic anxiety will kind of then lead to an episode of depression, and kind of chronic anxiety and rumination and that kind of thing, worry about things. You lose sleep over things you're worrying about, and then you start losing sleep. Then that may lead to abnormalities in the stress responsive system, and maybe energy metabolism, and this could all snowball. So that's another important aspect to this.
Well, and I think if we look at the way, I think one of the major challenges as it relates to something like depressive illness is how patients present. Even something like PTSD, how do patients present? If you just look at something like sleep, some patients have sleep abnormalities in terms of they can't sleep, and other patients always feel sleepy. If you look at things like body weight or food intake, some patients are overeaters and gain weight. Some patients are undereaters and lose weight. And so, I often think about it as sort of like Baskin Robbins, right? There's how many flavors of these disorders do we have? And I think, and I was talking to one of your other guests, Carrie Ressler, about this on one time, and we were talking about how I think this is the major obstacle that we have in effectively managing a lot of neuropsychiatric disorders and neurodegenerative disorders, is that we can't take this one box fits all approach. Is that the systems that are involved really depend perhaps on how the patient is presenting. And if we can identify how that patient is presenting, at least maybe that provides us some insight into which drugs might be more beneficial. And again, this is beyond what we've been talking about in terms of, those lifestyle changes. I think that's the groundwork, but layering on top of that in terms of the pharmacology, the major limitation that we're facing, in my opinion, is we don't know in terms of how a patient presents which system or which circuit or which peptide might be driving those pathologies. And if we had a better understanding of that, I think that would at least hopefully increase the efficacy of the existing drugs that we have and maybe identify some new targets for us.
Yeah. At Hopkins, where I have an appointment now. So we moved to Baltimore area in the year 2000. And of course I have a general practitioner I see once a year and so on. And so in the first years we were here, we had 30-minute appointments, right? And now it's down to like 20 or 15 or 20. So this is another big problem, that doctors, primary care physicians, aren't spending enough time with their patients, talking to them and asking them things about their sleep. Or, the patient will come in with some problem, and if there's a diagnosis of some specific disease or disorder, then a drug is prescribed. But that primary care physician, at least mine, never talks about diet, lifestyle, other things going on in your life, and all the major diseases that kill us, whether it's cardiovascular disease, cancers, neurodegenerative disorders, diabetes, kind of the risk factors, the diet and lifestyle risk factors are all the same for them.
Yep.
And there's just, patients never even have the opportunity to talk to a doctor that can kind of dig down into that. I'm from Rochester, Minnesota, where the Mayo Clinic is. And I went to Mayo High School. They have a high school named Mayo High School. And there are a lot of doctors' kids there, although my father was actually the prosecuting attorney for the county. But anyway, at the Mayo Clinic back then, all the doctors were on fixed salary. And they could spend as much time as they felt they needed with the patients. That's changed now. But it was like a really fantastic for the patients, right? Because I remember a kid, going in with my mom, or my mom had some issues or something, and the doctor would spend an hour with her, talking with her and going over things. So that's another big problem, I think.
Yeah, and it's one of the things that I talk with our students about. I do a lot of teaching with our second-year medical students, and we sort of dabble on the edges of healthcare issues. We tend to focus more on the basic science issues, but these are important issues as it relates to healthcare in general. Mm-hmm. And I think you could do an entire podcast just on that discussion, because when we think about as a pharmacologist, this new wave of drugs which we use now, which are very, very effective, these monoclonal antibodies, which target most often a particular cytokine, which are very effective in a lot of different immunological disorders, but they're incredibly expensive. So how do you balance, again, sort of that prevention aspect versus treating a patient now whose disease has progressed far beyond what you would like? And now the healthcare cost with treating that individual is going to be much higher than it would have been if we had done intervention strategies earlier on. But again, you might want to, that's a good idea for another podcast, Mark.
Yeah, I agree. Essentially, you alluded to inflammation there, right? These, for example, antibodies against tumor necrosis factor, which is kind of a classic pro-inflammatory cytokine. And I was going to ask this earlier on. So in these really nice studies where you selectively knock out the insulin receptor in neurons in the hippocampus and show that alone adversely affects the structure and function of the hippocampus. But with obesity, type 2 diabetes, there's kind of this pro-inflammatory, and in Alzheimer's disease, if you look at the brain, there's local inflammation. So essentially, I'm assuming your experiments then completely dissociated an effect of insulin signaling from inflammation, because that's another important feature of those experiments, right?
Right. So again, sort of teasing apart the cause from the consequence, like you mentioned, if you're looking at an obesity model, if you're looking at aging, if you're looking at those experiments where we administered the antisense into the hypothalamus, where the animals got fat, we did studies where we gave a leptin receptor antagonist, the animals got fat. And not just us, but you and so many other people have shown that in those settings with metabolic disorders, you have this increase in inflammation, and it's in the periphery. Now, we could have a whole other podcast on how that peripheral inflammation impacts the central nervous system, and that's one of the things that we're also looking at. But in terms of what's happening in those animals with the hippocampal specific insulin resistance, we see that those animals, we don't see any changes in inflammation. So peripheral inflammation in those animals is fine. HPA activity in those animals is fine. Peripheral oxidative stress is fine in those animals. So when you start looking at those, that's why I was thinking those are probably some of the causes: HPA axis dysfunction, inflammation, which is driving central nervous system insulin resistance,
that makes
which
sense.
then causes those other consequences. And of course, inflammation in and of itself is going to produce all of those neuroplasticity deficits. But again, one of the ways that might feed into those consequences is through insulin resistance.
I've had several podcasts where we touched on ketones.
Yeah.
Right. And so glucose is an important energy source for neurons, but they can also use ketones, which come from fats. And we never, I don't think we ever looked at the ketone transporter. It's called MCT. Monocarboxylic acid transporters move ketones into neurons, or lactate. There's actually some interesting work on that. So actually, we think I did a podcast with Steve Cunane, actually two.
Yes. Yeah, sure.
And he, his evidence suggests that, at least in mild cognitive impairment, neurons may be able to still use ketones pretty well, even though they're not. There may already be some insulin resistance. So that's another approach that's being pursued in terms of the energy aspect. Of course, insulin signaling in neurons, you mentioned you think it's increasing BDNF. So I think insulin's doing much more than just enhancing glucose uptake, and probably the insulin resistance is bad for the neurons, not just because they're not getting enough energy. But
so we actually did a seminar together. It was a symposium at last year's Alzheimer's meeting in San Diego, and he showed us some ongoing studies where they were able to demonstrate a very nice enhancement in cognitive function in patients with Alzheimer's disease on these sort of, the keto diets they refer to them as.
Yeah.
And, one of the criticisms of those keto diets has been that, okay, we're in a situation now where, how palatable is that for an individual, where you have this excess amount of ketones that they're taking, this excess fat. But what he showed is a very modest increase in ketones were able to effectively enhance cognitive function in patients with MCI and Alzheimer's. So I think that's very, very interesting early observations. And again, kind of feeds back to this idea about in terms of metabolism and how brain metabolism or deficits in brain metabolism probably play a really key role in neurodegenerative disorders.
Yeah, and well, the kind of, I think the study you're referring to, he gave the patients medium chain triglycerides, which
Right.
they're like immediate fatty acid precursors to the ketones. I'm not real keen on ketogenic diets for a lot of reasons, and again, that's a whole other podcast.
Right.
But it's possible to increase ketones even if you're not on like a strict, meat eating diet.
Right.
So, yeah. Okay. Well, let's see. I think we've covered pretty much everything, and I hope our viewers and listeners got some take-home message. A lot of times it's the same take-home message: exercise, don't overeat,
Right.
eat healthy food, avoid sugars,
Right.
keep your brain active. But you've also, your work and others in this area suggests that there's some hope for developing approaches to preserving brain function as people may be getting towards this kind of tipping point.
Yes.
A tipping point that may be sending them into cognitive impairment and Alzheimer's disease. Yeah, so people can kind of stay tuned. There's a lot going on. I think that's the encouraging thing. A lot of clinical trials. We'll see what happens.
Yeah, indeed. The tipping point, that's a great way to describe it. And I think, again, what we've really focused on in the last hour or so is, we need to be thinking before we get to that tipping point. But I do think, again, based on the discussions that we had at the Alzheimer's meeting last year, I think there's a lot of optimism. And I think it's going to take a multi-pronged approach. It's not going to be just one magic bullet, which is going to cure patients.
Yeah, and and it's also been in the press a lot lately in relation to Alzheimer's, the approval of the amyloid beta peptide monoclonal antibody as a treatment. The FDA approving it, even though the scientific advisory scientists who reviewed it recommended not to approve it. And there's been so many failures of this approach, the idea that you can just take an antibody and remove the amyloid and everything's going to be good. And that's clearly not the case. So and the reason I mention this, for years and actually several decades, the preponderance of funding for Alzheimer's research was oriented towards the idea that amyloid accumulation is the cause of the disease. Even though even a long time ago, and I'm a good example, there are many scientists in the field who said, hey, we don't think this is like the key thing. What about the mitochondria becoming dysfunctional? What about the oxidative stress? What about inflammation? What about this and that? And but the way the study sections were kind of organized, the people in them, a lot of amyloid studies got funded. Literally billions of dollars investment of drug companies went into it. And then the failures actually led to major drug companies like Pfizer discontinuing their research and drug development related to Alzheimer's disease. So it actually, that tunnel vision, not, as you were saying, not considering multiple things that we know are going on, and how do we kind of work on modifying all of those things. We know how we can do it, exercise, diet, but through pharmacotherapy.
Right. And I think for some patients that some patients aren't going to have the ability to exercise, especially if elderly populations.
Right.
So it's going to have to be a variety of approaches. But now you've got another podcast, Mark. You do one on healthcare perspectives from the pharmaceutical industry.
Okay, Larry, it's great to talk to you. I hope you have a good summer.
Thanks, Mark. Appreciate it.
Bye.
Bye-bye.